Here we go. You see it in presentation mode, or?
Yes.
Okay, great. So, well, thank you first for all of you for joining in. So I thought I would give you a few years ago, a couple of years ago, we started to discuss what would be next and discussing the idea of grand challenges. And we define a little bit what it would be. So grand challenges would be international projects. It would showcase metaproteomics. Why would we bother doing metaproteomics? It would address specific challenges or specific applications, and it would be open to everyone interested in participating. So we started to work on a few grand challenges, but the first question was, why metaproteomics? Why would we bother to do large scales or global scale type of project, and why not just use metagenomics? And I thought we would just put a slide there, and obviously if you're in the space, you're already convinced, but if you're not in metaproteomics, you might not be yet. But basically, if you look at this slide there, this is showing you three different individuals, and it's basically showing you what you would see in terms of if you did metagenomics in terms of predicting protein expression versus proteomics. So in the three individuals, 5 different treatment, but anything that is centered on 0 across the 3 individuals, metagenomics predicts what metaproteomics measure. And here I put on this graph 2 bars that are at log 2 1.5, which would be a 2.8-fold. So anything on the right of that bar and on the left of that bar is not predicted correctly by metagenomics. So that would mean on the right side, 37% for the first female is downregulated, but it's not predicted properly by metagenomics, or on the left side for all of them. And 2.8-fold change is a major change, a fold change difference in terms of looking at protein expression. So that means that if you go down to a lower fold change, it's even more significant what metagenomic is not predicting. And you can see that there are extreme at 20 and minus 20. And those extreme really means that metagenomics is totally off in terms of predicting the expression of proteins. So we cannot rely on metagenomics to predict what's going on in terms of protein expression, and proteins are the ones that are doing the work. And on top of that, you could add things like post-translational modification that are involved in the regulation and so on, that are important, that metagenomics is not going to predict. So the reason why we need metaproteomics and other omics is because we need to get down to that level of precision to understand the biology that is ongoing, the same way as it's been done in human. Now, if we look at the few grand challenges we defined, the first one was the dark field of metaproteomics, and this is related to this question. So 85% of the predicted peptides from the UHGG in terms of the MAG sequence are distinct if you predict them. There's only 16% that are common. But when you look at the observed results so far in metaproteomics, 79% are shared peptides, and they're not distinct peptides. So therefore, metaproteomics, we're measuring very few distinct peptides. And if you look in terms of what's going on, so metaproteomics is biased towards shared peptides, and we could go on and explain why, but don't have the time right now. Only a fraction of the MAGs have been observed by metaproteomics. That means that we're missing a lot of the microbes that are potentially out there. Many of the predicted peptides will not fly by mass spectrometry. That would explain some of those. And finally, many genes are not expressed. Not all genes that are in a bacteria or a microbe are going to be expressed. But you also see here on this figure is a little circle. So this little circle is, in fact, this circle there of all the peptides that have been observed put against all the possible peptides in the MAG. So it's a very small portion that we currently observe. Obviously, not all of them are going to exist, but nevertheless, by metaproteomics, we'll observe only a small portion, and many of those are shared peptides instead of distinct peptides. Even though we measure a small portion, it's still variable from a biological point of view. But where we want to get at in the first grand challenge is to flip this, to have way more distinct peptides. And distinct peptides are the ones that are rich in information from a taxonomic point of view. And to do that, we had proposed the human metapeptidome atlas, which would be increase the coverage of the distinct peptides and create an atlas. The outcome would be a database of observed distinct peptides that are annotated, which would be a very rich information to guide not only metaproteomics, but also the biology related to microbiomes. So this would be much more of a technological angle type of grand challenge. The second grand challenge is the functional diversity of the microbiome. So if you look at the world representation of MAGs that have been done so far, at least as of a couple of years ago, the UHGG database principally came from China, Europe, and North America. And what has been predicted through rarification is that we're expecting newer rare bacteria to be added, but not significantly increase into bacteria that are common across the globe. However, when you look at metaproteomics, as of a couple of years ago, most of it came from North America and Europe. Now there's an increasing amount of metaproteomics being done in China, and metaproteomics is far from being saturated. So that means that we do not know what are the proteins that are being expressed by the microbiomes around the world. So we don't know from a functional point of view whether the microbiomes are very different or not. So therefore, what we want to do is to be able to take what we know in metaproteomics and transfer it to be able to get a much better view globally of what's going on in terms of function. And that is important because understanding the function of microbiome is essential to be able to do, for example, intervention in human and other approaches. So why is the functional diversity of the metaproteome? What is the functional diversity of the microbiome across the globe? We don't know that. Is the functional diversity linked to metabolic output? Are post-translational modification of the microbiome conserved across microbiomes? We do not know that. Are PTMs linked to metabolite levels? Are the functional diversity and post-translational modification affected by diet and age? We don't know any of that, in fact. And are the functional diversity and post-translational modification linked to metabolite diversity? We don't, again, know any of that information. So, but can we observe functional diversity by metaproteomics? And we did a small exercise to verify that, because if we cannot observe it, there's no point to do a global scale project. But if you look at the peptide atlas that we published a couple of years ago, and if you compare North America versus Europe, you can see that there are species that were only, as of that date, only observed in North America and Europe. We're using 10 distinct peptides. And if you go with 50 distinct peptides, you can definitely see that there are common species, but some were only observed in North America and Europe. In fact, the literature on the genomic size that tells you that if you go down to strain level, there's about the overlap in strain level is very low. It's about 1% across individuals. So that means that functionally, there's a lot of value. There's a lot of value that hasn't been captured because we haven't sampled the world. So the grand challenge number 2 is the functional diversity of the microbiome. So the Human Metaproteome Project determined the functional diversity of the human microbiome. The outcome would be better understanding of microbiome functional diversity linked to metabolite productions and other functional output. And to do that, we need to define a set of microbiome samples that will allow us to reach saturation, just like the MAGs have been saturated in some space using mass spec approach such as DDA or DIA. And we need to enrich enrichment from these samples, and we need to have a reference microbiome, which as of yet doesn't exist. Finally, the grand challenge number 3, which is system mapping of microbiome functions and redundancy. Why is that important? Functional redundancy is a key determinant of the ability of a microbiome to respond to change. The more redundant a microbiome is, the more able it is to respond to a change, because it has different ways of producing the same output. Now, when the functional redundancy shrinks, losing a bacteria can be detrimental to the microbiome, because it's not able anymore to produce a metabolite, for example, that bacteria is producing, because it's out and it doesn't have something else to replace it. So if you look into prior studies, we've seen that, in fact, in disease such as inflammatory bowel disease, that functional redundancy is important, and it changes as the disease progresses from an inflamed to a non-inflamed region. That means that it's not just about composition, it's about the function that is being done within, in this instance, for example, in the terminal ileum, the function that is occurring there, and the ability for different bacteria to produce the same thing. And as that shrink, it seems to be linked to the disease. So therefore, the functional global output is important. If you look at it from another point of view, which is antibiotic resistance, for example, what has been shown is that the gut microbiome is, all of us has antibiotic resistance in our gut microbiome, but there's basically two populations. About 78% of the population has a basal level of antibiotic resistance, but there's another group, about 22%, that has 10 times more antibiotic resistance in their gut microbiome. I'm not talking about pathogenic bacteria here. I'm talking about commensal bacteria in the gut microbiome. And they have 10 times more antibiotic resistance, but also they have a lot of multi-drug resistance. Why is that important? You could say, well, in a recent study that Lurian did, what she showed is that, in fact, if you look at antibiotic protein resistance level versus functional redundancy, so if it shrinks, it gets worse. If it goes left, the microbiome is less functionally redundant. It's totally linked to the strong correlation for 115 drugs between the level of antibiotic resistance being expressed and the change in functional redundancy. Now, if you look at across all of those, the basal level of that individual 45 is 1.2%. So without any drug treatment, without anything, there's 1.2% of the proteins that are antibiotic resistant in that individual. This 1.35, this 1.8, this 1.5, this 1.45, this 1.1. So if you it should be able to see a pattern there. The higher the initial level of the microbiome of antibiotic resistance, the stronger the correlation between the level of antibiotic resistance expression increase versus the functional redundancy. That means that if you have a strong basal antibiotic resistance, treatment with drugs boosts the antibiotic resistant protein level, and then the functional redundancy of the microbiome shrinks. But it's not the same if you have very low level. This would not be apparent at all without looking at proteins. So therefore, it's important to look at proteins, but it also tells you that individuals are very different, and that antibiotic resistance also plays roles in how the microbiome respond at the functional level. So it, again, brings the idea of the grand challenge number 3. I think it reinforces the reason of why to do that is to systematically map microbiome function and redundancy. Why 70% of UHGG species have not been cultured, 40% of UHGP don't have function, and we have limited understanding of functional interaction across species, limited understanding of compound microbiome interaction. I just showed you antibiotic resistance now being important for compound microbiome interaction. So therefore, what we're proposing to do is to do the Human Microbiome Systems project. There's to determine functions, network, and interaction in the microbiome, and the outcome would be better understanding of microbiome systems. And to do that by challenging microbiome with different compounds. And that can be done through different assays, but it's now possible to take microbiomes, challenge them with compounds, and measure the outcomes. And you can do this at the systems level. And this will give us a better understanding of how microbiome responds to challenges, which is really what they systematically do, and understand the mechanism involved in those, and understand the regulatory processes involved in those. And most of that is decided through proteins, their post-translational modification, and a response to presence of antibiotic resistance, for example, and so on. So those are spaces that can only be done by looking at the metaproteome. That's why we've been putting those forward. The project will evolve as the science move, but those are places that metaproteomics would very likely have tremendous beneficial outcomes from the point of view of biology. And that's a quick overview. Thank you.